Hey everybody, thank you so much for being here today. I hope that I don't let you down by not being Yogesh. This is just a project that we conceived last year to try to explain essentially perturbation responses in a way that is, simple and interpretable. And so essentially what we found out is that, and this should not surprise lots of people in this room who are physicists, but fluctuation structure in the unperturbed state turns out to predict genome-wide perturbation responses. And so why do we care about perturbation responses? Well, I think that we do all care about them, but we can view cell state transitions as being responses to transcriptomic perturbations, whether we're dealing with stem cells turning into differentiated cells, um, or reprogramming of stem cells, viral infection, directed evolution, or cancer resistance or metastasis. These are all transitions where we can hope to figure out, causal transcriptomic features that, when perturbed, can actually influence these phenotypes. And so this is a difficult problem because most of the phenotypes that we actually care about are not single gene Mendelian traits. In fact, we have to make many to many phenotype-genotype maps in order to describe them. And so I'll just first go over a little bit of the history of these genetic perturbations, which started with knockouts in the 70s. You knock out a gene and suddenly your mouse becomes a big mouse. Or you CRISPR a gene and suddenly your flies go from having gray eyes to red eyes, for example. And there are a number of different techniques in order to do this sort of gene editing or perturbations. But I'm going to focus on CRISPR, and I'm going to focus on CRISPR-I and CRISPR-A. So CRISPR that does not completely knock out a gene, but just changes its expression. So CRISPR-I will knock down a gene, CRISPR-A will overexpress it. And so up until recently, we haven't been able to really do this en masse. It actually took quite a lot of work to knock down the gene. Upon the advent of pooled CRISPR screens, where we can essentially transduce a large library of guide RNAs into a population of cells, grow them up, sequence them, and because each cell has a barcode telling us which perturbation it harbors, we can actually start to get a sense of ground truth as to which thing was actually perturbed that caused the phenotype that I care about. And so the problem with this is that it's great, but you traditionally have to have a different screen for every trait, and you have a combinatorial explosion of different perturbation possibilities. So if you want to perturb every gene in the genome, that's great. That's on the order of 10,000 genes. If you want to perturb every 5 genes in the genome, you just can't do that. And so up until now, there have been a host of different models looking primarily in the forward direction, meaning that we want to predict perturbation response given perturbation. And these models fall very broadly into 2 classes: linear models, which are basically regression models, and AI models, which are great for prediction, but sometimes not as interpretable. And so today I'm going to be talking about a perspective of a very, very simple model in order to predict how genes respond to perturbations, and in order to predict which perturbations those genes are actually responding to in the first place. So essentially, an overview of the model is this. We take a bunch of perturb-seq data, which again constitutes a bunch of individual CRISPR perturbations. Since we know which genes were perturbed in which cells, we can start to actually look at, for each perturbation, the distribution of control cells versus perturbed cells. And at the end of the day, the model basically says that that change in gene expression can be recapitulated by the covariance of that perturbed gene with all other genes and a vector of quote-unquote perturbations. So if I know what gene was perturbed, I have a single U in there, all the other U's are 0. And I have a single fitting parameter that basically tells me what do I scale my correlations by in order to actually, fit response. And you might be saying that model is way too simple. but it turns out to actually work to some extent. So essentially, what I'm showing you here is just a diagram of a bunch of different perturb-seq data sets. These are real data sets in a variety of different cell types and different CRISPR modalities in order to basically test whether the co-fluctuations of genes are important as opposed to just the variances of the genes. We made a mean field model where we basically scrambled the off-diagonal correlations but kept the individual distribution of genes the same. And we see, lo and behold, for all of these individual perturbations, that we can get very, very high R-squared scores, compared to the mean field model. And so how am I actually doing this? Well, I basically want to minimize I want to do a least squares problem on the distance between my delta x, my perturbation response, and my sigma u, my predicted response. And the way that I do that is just analytical there. My u star is given by the inner product between the column of the the column of the correlation and my delta x over the magnitude of that column. And so you might be saying to yourself, well, you just use the answer to get the answer. But it turns out that since this constant is the same for every single gene that's perturbed, you can just hold out a bunch of genes, fit some of the genes with the constant, and then test on the other set of genes. And so it turns out that we can actually do this pretty well. And I'm showing estimator convergence, basically showing how many genes I have to use in order to get a steady Pearson correlation. And remarkably, it turns out that, even though we have tens of thousands of genes, we really only need on the order of 10 in order to get a good fit. So this is sort of what the fit looks like if you look at delta x versus the gene index. I picked a couple of, good fitting genes, but you may have a hard time telling the difference between the, experiments and the predictions. We also wanted to see, how stereotyped are these correlations in primary cells. It turns out that they're stereotyped a non-negligible amount. What we wanted to do was predict the response of neuron cells in the lab using correlations from primary cells from cell atlases. And so we did that with the brain atlas, with the lung atlas, and with the intestine atlas. And what we see is that, what you might expect, the brain atlas, which has a bunch of neuronal cell types, does better at predicting how neurons respond. But what was pretty interesting is that the cell type in the intestine, for example, that does the best at response, even though it's not nearly as good as like real brain neurons, is the neuron, the intestinal neuron. And so we have a sense that these are actually stereotyped. So what I've showed you so far has been the forward problem, predicting response. But I would argue actually that the more interesting problem is the inverse problem. Given a response in a correlation matrix, how do you predict which genes were perturbed in the first place? If we knew that, then we could start to combat some of these complex diseases. And so we do this in a Bayesian sense. We basically add a little xi over there, which is normally distributed. Using central limit theorem arguments, we can figure out what the actual covariance of that likelihood is. It turns out just, yes, Aditya?
So if I understand correctly, you're looking at fluctuations from the perturbations, right? So you knock up a bunch of genes or you knock down a bunch of genes and you look at co-fluctuations of...
well, actually, we're looking at fluctuations just in the control cell population.
Oh, I see. So just sort of stochastic fluctuations. So you're not actually doing the perturb-seq yet.
So we use the control fluctuations to predict what the perturb-seq
I see. Okay.
would give us.
Oh, okay, I understand. Okay, thanks.
Okay. And so with this Bayesian approach, for each gene, for each perturbation, we can basically say, what's the probability that that gene was really perturbed? And we can look at the effect sizes that we get to basically infer whether that gene was knocked down or activated, overexpressed. And so it turns out that I'm showing you an example here from double perturbations. So we took true perturb-seq data that was doubly perturbed, and we were able to pick out, using this Bayesian model, the constituent single genes that were perturbed in that double perturbation. And so that's, I would argue, pretty strong evidence that we can actually, pick out causal genes. You might be asking yourself, well, can we do this with drugs? And the answer is yes. So using a very analogous approach, where instead of looking at each individual gene, we basically look at each drug and get a transcriptomic response from that drug, we can then go to our library of drugs and ask, which one has the response that looks most similar to the response of this drug that I don't know. And so we can, at the end of the day, accurately infer what drug a population of cells got, and we can do so handedly better than just using like a log fold change, for example. So what I've told you about are, synthetic systems, but we're really interested in predicting drivers, for example, of drug resistance in cancer. And so what I'm going to do here is basically treat the untreated naive cancer cells before drug as being the control cells. I'll look at how they fluctuate and try to predict essentially what's causing the resistant colonies. And so when I do that, I get essentially a list of genes. I can weight them by how important the posterior model says that they are. And this is all, good and cute, but it doesn't tell me if the model actually works. So we went into the lab and we actually, and I should have acknowledged Nitu, who's a wonderful postdoc in our lab. We went to this lab and we actually knocked down a bunch of these genes that we predict should actually cause resistance before we treated with drug. And indeed, we see, a lot of the time, 10 out of 11, hits out of the top 200, we see that the incidence of resistant colonies dropped a lot.
Which cell line is it?
So this is a melanoma cell line, BRAF melanoma. Actually, no, sorry, this is Miopaca. This is a KRAS pancreatic cancer. Yes? Yes?
So it's predictors of resistance. This is the way that I think about that is if you knocked it out, you got a lot more growth under the drug. You're saying it's like a synthetic lethal. You put the drug on them, that this then causes them. If you knock this out as a pair, they die more often. So it's a sick relationship rather than a knock this out and it drives increased fitness.
So, I mean, the way that I sort of think about it is that there's a subpopulation of cells that may be predisposed to resistance, and those subpopulation of cells might have, genetics or gene expression signatures that are indicative of resistance, like causing resistance. That's what I think that we, somewhat pick up on. And so if we predict that a gene, causes resistance, we can knock it down before we treat with drug and hopefully see that a lot fewer resistant colonies emerge.
So, yeah. but it is a sick relationship. Basically, what I'm saying is like it's a down. This isn't an up assay saying if I knock this out, I get huge increase in resistance of the cells.
No,
in the population.
I don't believe so. No.
Yeah, because that's why I was expecting that. And then I saw these results. I was like, that's not what that is.
But we have tested again with like non-targeting and scrambled controls, which indicate that the cells with the guides are actually growing the right way. Mm-hmm. So it's not just that we put guides in there.
Oh yeah, no, I did get that.
Okay. Yep. Sorry, I forgot.
No, I'm confused. So the causative genes that you identify are the one that you measure after the perturbation, right? But you cannot take into account the diversity of cell states before the perturbation and which cell give rise to which cell after. Is that correct?
Because
on differently.
I definitely agree with that, but we're somewhat taking that into account by using the fluctuations of the naive cells, I believe.
That's your Bayesian approach.
Yeah, so that is essentially what we do here.
Yes, but you destruct the cells. There are two different cell populations, right?
Yes.
Yeah. Okay.
Yes, that's right. And so now I'll just sort of go into a little bit of what I view as the future of this work. So now that we've basically found that these correlations are somewhat stereotyped in primary cells, we're pretty interested in looking at, are they also stereotyped across, evolutionary history? And if they are or aren't, how does that change with, the length along the tree that you are from, human cells? We're also interested in looking at this from a clonal perspective. If we actually barcode cells, are fluctuations in a distant relative indicative of responses to perturbations of a progenitor. And in the future, we'd like to really integrate these sort of models with neural networks and try to essentially, constrain their output or make them more interpretable, using these sort of simple models. And so as just a little bit of a teaser, we basically thought to try to use this to predict, what cell fate cells would have. So we're using here the Larry data set, I think from Ellen Klein's lab at Harvard. And essentially, the name of the game is that we have a population of undifferentiated cells, and they differentiate into cell types with some probability, right? And so if we look at what those probabilities actually are for all the undifferentiated cells, we see that we get a fairly high Pearson correlation. And if we are interested, again, in basically just what the final future lineage is, we can predict that as well. And so, in summary, we essentially developed a simple linear response framework in order to predict both how gene fluctuation can produce perturbation response and how a given perturbation response can be attributed to a finite number of gene perturbations. And so with that, I'd like to really thank everybody in my lab, particularly Yogesh, as well as Akshit Goyal and Suri, who have been, really amazing in this project. So with
few minutes, do you want to talk about the normalization point? Because Leo,
yeah, I would love to.
maybe that's
So I'll talk about the normalization point. I don't really have any slides on this, but
we'll believe
but yeah, so we believe that at least for this sort of approach, the best way to normalize your data is to not normalize it. So what I mean by that is that you just take raw counts. Obviously, there's going to be some sequencing depth artifacts there, but we think that what we see empirically is that once you normalize the data, the signal drops a lot. And so why is that? Well, I think that's mainly because when you normalize data, you're literally projecting out the total mRNA component. And some perturbations just happen to have more mRNA. And that is a biological feature, not something that you should throw in the garbage.
correlations.
And also, yes, so normalization introduces a lot of spurious negative correlations that are not there in reality. And so if you were to, go and say, and in this sort of model, correlations are literally the interactions. I don't believe that they're, literally interactions, but yeah, you're throwing away a lot when you normalize. Yes, Katie?
Can you talk about how much of a determinant feature the amount of RNA is? Because it sounds like this is something we've been wondering about.
Yeah, so.
And I'm just wondering, how do you...
Sorry.
The question is like, how much is the total RNA? Sorry.
Yeah. So Katie asked essentially, how important is the total mRNA in the cell? To what extent is it a determinant of perturbation response? And so we think that, it really depends on the data set and the cell line, but one of the features of this theory that we've built is that a lot of these responses are channeled through a number of small directions, like soft modes. So basically, there are directions in gene expression space that are easier to push on, and that most of the perturbations will be channeled through those directions. Essentially, when you normalize, you're not only suppressing those, but you are, again, getting rid of your total mRNA component. You can actually model that, and you see that for some perturbations, it doesn't really matter a lot. For some perturbations, the signal is totally destroyed. So it can be important, yes. Yes, Sean, sorry.
So when you pick up these genes from this analysis before you've added the drug, the perturbation, how much of it sort of matches this notion of memory genes and things that are inheritable over long time scales?
That's a very good question. We honestly haven't looked at that too much, but I would be very happy to talk about it. And
Look into it.
Yeah, we should look into it.
This metric, so we should talk, but these the fluctuations you're thinking about here, you have no sense of timescales in which these sort of decay.
Well, I mean, from the perspective of that, near bifurcation correlations are really the same as the Jacobian, so their eigenvalues dictate timescales. We do have a sense that some of these data sets have an extreme separation of timescales, which makes them very predictable. But yeah, and I feel like we're sort of running out of time. So okay, Jose.
Why covariance is enough? Why not higher order terms?
Yeah, that's a good question.
We had a good reason for that. But here you could have like multiple genes coming together. Is that you don't have enough data? Or that's reality?
Yeah, so I would say in reality, if we could converge higher order moments of the gene by cell matrix, then I would suspect that we would do a better job. But in a lot of these cases, again, because you have these soft modes and like really only a couple directions that matter for perturbation propagation, it turns out that in that case, the Jacobian, which really does determine unequivocally perturbation response, reduces to the correlation matrix in that case. Gabe?
Lovely as always been. What will happen with multimodal?
Yeah.
Like when your perturbation or your original cell population has multimodal structure, does this technique work?
Yeah, that's a really good question. So we have reason to believe that actually near a bifurcation it should work actually extra well. For if only because in that case you basically get like a divergence in the gap of your spectrum. And so really only a single direction matters, and that direction, is the same for every gene essentially. Yes, Shin?
Yeah, so this relationship you have, that's sort of the fluctuation response theory in the equilibrium.
Yeah.
Yeah, 100%.
Exactly. That's my concern.
Yes. Okay. So,
but this is a steady state. Are you sure this is in the equilibrium?
I am not sure it's in equilibrium. In fact, I would bet you it's not. But there is a separation of timescales, I believe, between the binding and unbinding events of the the transcriptome and the actual overall phenotype, which occurs on the order of like days or weeks.
If I can add one small comment to what Jin asked just now. So, there is a relation you can write down, the Lyapunov relation between the Jacobian and the correlation matrix that is valid in a steady state that doesn't have to be equilibrium. And when all of these results, one way to explain them after the fact is that the Lyapunov relation ends up simplifying dramatically in some cases where the rank of the matrices are low. And that's sort of what we think is happening behind the
general, there should be another term.
Sorry, what?
In general, there should be another term. Response is not just purely fluctuation.
Yeah, certainly there's a circulation term that we're neglecting. Oh, sorry.
That's what.
That's small.
So we can chat about it in the
okay.
Yeah.
I don't think that's true. It's just a tilting.
Thank you.